Hello, my name is Peter Jermain. Today I'm going to talk about a novel optical approach for cancer detection at the cellular level. The title of this dissertation is Quantitative Optical Cytopathology for Cancer Diagnosis. Here is an outline of the presentation. To begin, I would like to provide some background information. Fluorescence is a physical phenomenon that occurs when an incident photon is absorbed by a fluorescent molecule, exciting an orbital electron to a higher quantum state. Various non-radiative transfer mechanisms serve to dissipate a fraction of the absorbed energy. The electron remains in the excited state for a duration called the fluorescence lifetime, prior to emitting a fluorescent photon. Due to the range of potential non-radiative interactions, the spectrum of fluorescence emissions is composed of a distribution of wavelengths. In general, fluorescence photons have lower energy and longer wavelength relative to that of the incident photons. Fluorescence polarization is a widely used optical technique that characterizes degree of polarization of fluorescence emissions with respect to the polarization state of the excitation light. It is defined by the equation shown on this slide. It's equal to the ratio of the difference between the copolarized and cross-polarized intensity divided by the sum of the co- and cross-polarized intensity. It is a ratiometric quantity that doesn't depend on the absolute intensity of the fluorescence emission. What's interesting about fluorescence polarization is that it is sensitive to the local environment of the fluorophore, its binding, and fluorescence lifetime. One factor that influences fluorescence polarization values is binding. An unbound fluorophore is able to rotate rapidly under the influence of Brownian motion, and the observed polarization of the fluorescence emissions will be lower, whereas a bound fluorophore exhibits less rotation during the lifetime of the excited state and the fluorescence polarization is higher. Another factor is viscosity. A fluorophore embedded in a low viscosity medium exhibits faster rotation, which causes decreased polarization of the fluorescence emissions. Whereas a fluorophore in a high viscosity medium shows slow rotation, which preserves high fluorescence polarization values. Our group is exploring the fluorescence polarization of an exogenous fluorophore called methylene blue, which you will see abbreviated throughout this presentation as MB. It is a positively charged phenothiazine dye that has some of the important physical properties listed on this slide. Absorption maxima occur at approximately 609 nanometers and 668 nanometers. Peak emissions occur between 670 to 710 nanometers. And the fluorescence lifetime of methylene blue in water is relatively short, at 0.5 nanoseconds. It is a widely used cytological stain that is already approved by the United States FDA for human use. Previously, it has been shown that methylene blue can enhance the contrast of cancerous lesions in reflectance and fluorescence images. We are exploring methylene blue fluorescence polarization for cancer detection. Some previous work that has been conducted in this area includes the following. Wide-field imaging of methylene blue-stained skin cancer sections was used to identify non-melanoma skin cancers ex vivo. Methylene blue fluorescence polarization was used to delineate breast carcinoma margins. It was used to map non-melanoma skin cancers in vivo during surgery. And more recently, this technique has been used to distinguish between cancerous and normal breast cells in human cell cultures. That brings us to this dissertation, which includes the first two studies of breast and thyroid cells obtained from the actual patients. This work utilized fluorescence polarization imaging, a simple and robust technique. Here's how it works. Linearly polarized light is used to illuminate the sample. The copolarized and cross-polarized fluorescence emission images are obtained, and these are used to calculate the quantitative fluorescence polarization image that provides the value of fluorescence polarization for each pixel. The type of imaging we employed is called confocal microscopy. This technique uses point source illumination and point-wise detection. An objective lens focuses light onto a small volume at the focal plane. Light from the focal plane is sent towards the detector by a dichroic mirror. A spatial pinhole positioned at the conjugate focal plane serves to reject the out-of-focus light, so that only signal from the focal plane is registered by the detector. This slide shows a diagram of the multimodal imaging system used at the Advanced Biophotonics Laboratory. As mentioned, the fluorophore we're using is methylene blue. It's excited at 642 nanometers, and fluorescence emissions are registered in the range between 670 to 710 nanometers. The system provides 3 detection channels, including reflectance, copolarized fluorescence emission, and cross-polarized fluorescence emission. When we use the 63x objective lens, the field of view is 200 microns squared. Lateral resolution is better than 0.9 microns, and axial resolution is about 2 microns. The reflectance and fluorescence images display cell morphology, whereas fluorescence polarization images provide the quantitative fluorescence polarization signals for each cell. The confocal microscope exhibits bias in the transmission of different polarization states of light. Therefore, we used a system calibration coefficient called the G factor that characterizes sensitivity of the detection channels to polarization in order to calculate accurate fluorescence polarization values. In order to do this, 2 solutions of methylene blue were prepared. One had the dye dissolved in phosphate buffered saline, and the other in glycerol. Fluorescence polarization values of both solutions were measured using the confocal microscope. Fluorescence polarization values were determined using vertically polarized excitation and horizontally polarized excitation. Fluorescence polarization is a property of the sample and irrespective of the instrument that was used to measure it. Therefore, by choosing samples of known fluorescence polarization and solving, we can calculate the system G factor, which was determined to be 0.75 independently for both solutions. To verify the correct value of G, fluorescence polarization measurements of both solutions were compared with measurements from a commercial spectrophotometer. Once we captured images of cells using the multimodal imaging system, the images were processed as follows. Fluorescence polarization data was processed using Metamorph software. The copolarized and cross-polarized fluorescence emission images were thresholded to remove background and saturated pixels. The cells were manually segmented, and the fluorescence intensity values of each region of interest were used to calculate the fluorescence polarization value, using the formula shown here, which includes the G factor. The fluorescence polarization value of each cell was obtained by averaging over 3 measurements. Fluorescence polarization images were generated using a custom MATLAB code, and those images were pseudo-colored on the basis of pixel intensity to display the fluorescence polarization signal. Now I would like to transition and present results of the first study presented in this dissertation titled Translational Potential of Fluorescence Polarization for Breast Cancer Cytopathology. The motivation for this project is that each year more than 300,000 new cases of breast cancer are diagnosed in the United States. The standard of care for diagnosing breast cancer is hematoxylin and eosin histopathology, which is expensive, labor-intensive, and time-consuming. It relies on subjective visual evaluations of cell and tissue morphology to diagnose the disease. An alternative approach called fine needle aspiration, or FNA for short, offers a faster, more minimally invasive method to diagnose breast cancer at the cellular level. However, FNA interpretation is prone to error as it relies on visual assessments of cell morphology, for example, the nuclear size and shape and mitotic activity. Diagnostic sensitivity of FNA ranges between 35 to 95%, and false negative rates up to 40% have been reported. In addition, FNA provides no information regarding the tumor grade. Previous work at the Advanced Biophotonics Laboratory demonstrated that fluorescence polarization of methylene blue is significantly higher in malignant breast tissues as compared to normal. Another study from our group showed that fluorescence polarization of methylene blue is significantly higher in cultured breast cancer cells as compared to normal. That study also sought to explore some of the mechanisms behind increased fluorescence polarization in cancer. It demonstrated that the higher fluorescence polarization values in malignant cells are associated with shorter fluorescence lifetime of methylene blue in cancer relative to normal. It also demonstrated that elevated fluorescence polarization values are associated with greater uptake of the dye in the mitochondria of breast cancer cells as compared to normal. Several types of malignant cells, including breast and thyroid cancer cells, are characterized by elevated negative mitochondrial membrane potentials. Reports indicate that cancer cells with elevated mitochondrial membrane potentials preferentially attract positively charged molecules like methylene blue. The mitochondrial matrix is more viscous than primarily aqueous cytoplasm. Therefore, our hypothesis is that greater binding and accumulation of the dye in mitochondria of cancer cells could result in increased fluorescence polarization values, as it restricts rotational mobility of the fluorophore during the lifetime of the excited state. The previous studies established feasibility of fluorescence polarization for cellular level cancer detection. However, cultured cells are grown under well-controlled experimental conditions, and they provide only a simplified model for cells obtained from clinical aspirates. Therefore, this study investigated whether fluorescence polarization of methylene blue can be used to differentiate cancerous and normal cells in breast FNA samples. The breast FNA specimens were obtained from freshly excised, pathologically diverse breast tissues following surgery at the UMass Memorial Medical Center. The samples were transported to the Advanced Biophotonics Laboratory at UMass Lowell, where the cells were plated in glass-bottom petri dishes, stained in 0.05 milligram per milliliter methylene blue solution for 20 minutes, and imaged using the multimodal confocal microscope. The optical data were analyzed and compared to clinical histopathology and cytopathology assessments. Statistical analysis was performed using a mixed effects model to evaluate the significance of differences between cancerous versus non-cancerous cells and between the different breast cancer tumor grades. In total, we imaged and analyzed 44 breast FNA specimens collected from discarded breast tissues of 28 female subjects between 20 to 87 years old. This table shows the detailed clinical information and fluorescence polarization data for the 19 malignant samples, which included 15 invasive ductal carcinoma and 4 invasive lobular carcinoma. This table shows the corresponding information for 10 benign samples, including 6 fibroadenoma and 4 intraductal papilloma, as well as the 15 normal specimens. For statistical analysis of results, we grouped the cells into 3 diagnostic categories, including all malignant, all benign, and all normal cells. We found that fluorescence polarization of methylene blue was significantly higher in the malignant group versus the benign or normal groups. We also looked at the 5 histological groups and found that methylene blue fluorescence polarization of ductal carcinoma and lobular carcinoma samples were significantly higher relative to the fibroadenoma, intraductal papilloma, and normal samples. This scatter plot shows the fluorescence polarization value versus cell size for all 3,808 cells in the study. The cell sizes were determined from the dimensions of the manually segmented regions used for analysis. Each marker corresponds to an individual cell. Cells in malignant samples are represented by red triangles, cells in benign samples as blue circles, and cells in normal samples as green diamonds. We can see that malignant cells tended to exhibit higher methylene blue fluorescence polarization values versus the benign or normal cells. It should be noted that no cell in a benign or normal sample had a fluorescence polarization value above 0.233. This slide shows quantitative assessments of 5 representative samples, one from each histological category. The top row shows fluorescence polarization images that have been pseudo-colored on the basis of pixel intensity value to display the magnitude of the fluorescence polarization signal as given by the color bar, where black represents a value of 0 and red represents a value of 0.4. As can be seen from the images, cancerous cells in panels A and B show distinct contrast relative to the non-cancerous cells in panels C, D, and E. For example, the ductal carcinoma and lobular carcinoma cells appear as yellow and red colors because they have higher fluorescence polarization signals versus the fibroadenoma, intraductal papilloma, and normal cells, which have lower fluorescence polarization values and appear as green and blue in color. In the second row, we can see scatter plots that display the distribution of cellular level fluorescence polarization values for all of the cells in each sample, as well as the mean fluorescence polarization value of the specimen, as given by the horizontal yellow or green line. The scatter plots are important because they demonstrate that quantitative assessments can facilitate diagnosis based on fluorescence polarization values, irrespective of the images. Statistical analysis revealed correlation between fluorescence polarization values and breast cancer tumor grades. The grade 2 tumors exhibited significantly higher methylene blue fluorescence polarization versus grade 1. In addition, fluorescence polarization of grade 2 was significantly higher than grade 3, which is consistent with reports of decreased mitochondrial content and numbers in the grade 3 tumors. The fluorescence polarization images were processed from fluorescence emission images, which can be utilized to provide additional diagnostic information on cell morphology. On this slide, the top row shows representative methylene blue fluorescence emission images that correspond to fluorescence polarization images that we saw 2 slides ago. The fluorescence emission images display cytomorphology, for example, eccentric nuclei in the lobular carcinoma sample and smudged nuclei in the intraductal papilloma sample. These features are similar to the features displayed in the clinical cytology images in the bottom row. To summarize the breast FNA study, fluorescence polarization of methylene blue was significantly higher in cancerous versus non-cancerous breast cells, The cellular level fluorescence polarization values were not specific to each patient. Fluorescence polarization images provided quantitative contrast between cancerous and non-cancerous cells, whereas the fluorescence emission images displayed morphological features comparable to cytology. Statistical analysis revealed correlation between methylene blue fluorescence polarization values and breast cancer tumor grade. Now I would like to transition and to present the results of the second study included in this dissertation titled Fluorescence Polarization Imaging of Methylene Blue Facilitates Quantitative Detection of Thyroid Cancer in Single Cells. We were interested to apply the fluorescence polarization method to thyroid cancer because the standard of care for detecting thyroid cancer is fine needle aspiration cytology, which requires analysis at the level of single cells, including subjective visual assessments of cell morphology to identify the disease. Fine needle aspiration cytology results depend largely on the skill and experience of the reading cytopathologist. As a method, it is labor-intensive, time-consuming, and expensive. This area represents a significant challenge for the healthcare system because most adults will develop one or more benign thyroid nodules during their lifetime. The American Cancer Society estimates increasing incidence of thyroid cancer with about 44,000 new cases diagnosed annually in the United States. The Bethesda system for reporting thyroid cytopathology is used by pathologists to categorize thyroid FNA specimens as cancerous versus non-cancerous. What's important to know is that there are 6 categories. Category 1 is non-diagnostic or unsatisfactory samples. Category 2 are the clearly benign lesions, whereas Category 6 are the clearly malignant lesions. Categories 3, 4, and 5 are the cytologically indeterminate categories, meaning that the cells cannot be reliably classified as cancerous or non-cancerous. Overall diagnostic accuracy of the Bethesda system is low when including lesions from the indeterminate categories. Objectives of this study included investigate feasibility of using methylene blue fluorescence polarization imaging to accurately sort the thyroid cells into correct malignant versus benign or normal categories, and also to evaluate methylene blue fluorescence polarization for the discrimination of cytologically indeterminate thyroid nodules. This was a double-blind study, meaning that researchers from the Advanced Biophotonics Laboratory were blinded to the histopathological diagnosis at the time of imaging and data analysis, and the study pathologists were blinded to the optical imaging results. Cells were scraped from freshly excised thyroid nodules or adjacent normal thyroid tissues and transported to UMass Lowell. At the Advanced Biophotonics Laboratory, the cells were plated in glass-bottom petri dishes. Erythrocytes were lysed, and the cell layers were stained in 0.05 milligram per milliliter methylene blue solution for 20 minutes. Optical images of the cells were acquired, processed, and compared to the findings of clinical histopathology. Results were statistically analyzed using a mixed effects model. Following the imaging experiments, a trypan blue exclusion test was performed to evaluate cell viability. All of the cells which had previously been stained with methylene blue were stained using 0.4% trypan blue and counted under a light microscope. Trypan blue only stains dead cells. Therefore, live cells appeared blue, whereas dead cells appeared purple because they were stained with both methylene blue and trypan blue. Using this method, we determined that cell viability after imaging was greater than 95%. In total, 32 samples were obtained from 21 subjects, including 19 samples from the UMass Memorial Medical Center and 13 samples from Lowell General Hospital. These included 13 malignant samples, 7 benign cases, and 12 normal specimens. The malignant and benign samples included 12 that were categorized as cytologically indeterminate. This slide shows representative multimodal optical images of cancerous and benign cells. The top row shows fluorescence emission images, panels A and B. The fluorescence emission images display cytomorphology, for example, subcellular features such as the nuclear size and shape. These are the types of visual features that pathologists train for many years to identify under the microscope. It is a challenging process because oftentimes benign cells present morphology that mimics features of carcinoma. In order to help pathologists examine the cells, we digitally stain the fluorescence emission images based on pixel intensity to appear like Papa-Nicolau stain, with cell nuclei blue-purple and cytoplasm blue-green in color. The corresponding pseudo-colored fluorescence polarization images in the bottom row, panels C and D, provide a quantitative measurement for each cell. Here, the pixel values represent the magnitude of the fluorescence polarization signal, as indicated by the color bar. This slide shows additional multimodal optical images of thyroid cells. Importantly, these 3 specimens were indeterminate on cytopathology, meaning the pathologist could not reliably classify any of the samples as malignant versus not malignant. Fluorescence emission images are in the top row, and fluorescence polarization images are in the bottom row. The 2 columns on the left are cancerous samples, papillary carcinoma and follicular carcinoma, whereas the column on the right is a benign sample, follicular adenoma. What I would like to highlight here is that while the fluorescence emission images show subtle differences in morphology, the fluorescence polarization images display clear quantitative contrast between the cancerous and benign cells. It's easy to see that the cancerous cells appear in yellow and red colors. They exhibit higher methylene blue fluorescence polarization values than non-cancerous cells, which appear in blue and green colors. So this is a case where optical imaging assessment could help reduce diagnostic uncertainty. For statistical analysis, samples were organized into 3 diagnostic groups, including all malignant, all benign, and all normal samples. Methylene blue fluorescence polarization was significantly higher in the malignant group versus benign or normal groups. Samples were also organized into 6 histological groups, including medullary carcinoma, papillary carcinoma, follicular carcinoma, follicular adenoma, multinodular goiter, and normal. Fluorescence polarization of methylene blue was significantly higher in malignant samples for all of the cancerous versus non-cancerous sample comparisons. Please note that the single medullary thyroid carcinoma sample was not included in the statistical analysis. Finally, and most importantly, analysis of 12 cytologically indeterminate samples revealed that fluorescence polarization in the malignant nodules was significantly higher than in the benign nodules. This slide shows another interesting result from the study. The scatter plot shows fluorescence polarization versus cell size for all 3,521 cells that we analyzed. Red triangles represent malignant cells, dark blue circles are benign cells, and light blue squares are normal cells. There is pronounced overlap of cancer cells at higher fluorescence polarization values, and similar overlap of non-cancerous cells at lower values, indicating that the fluorescence polarization values are not specific to each patient. No cell in a benign or normal sample had a fluorescence polarization value greater than 0.245. To summarize the thyroid cell study, both cancerous and non-cancerous thyroid cells remained viable during and after the imaging experiments. Thyroid cancer cells exhibited significantly higher methylene blue fluorescence polarization values versus benign or normal cells. This method accurately discriminated cytologically indeterminate thyroid samples into the correct malignant and benign categories. Methylene blue fluorescence polarization and fluorescence emission images facilitated quantitative and morphological assessments of the specimens, respectively. Now that the results of the breast FNA study and the thyroid cells study have been presented, I would like to discuss some general conclusions. Methylene blue fluorescence polarization was significantly higher in malignant cells versus benign or normal for both the breast FNA samples and for the thyroid cell specimens. Advantages of using fluorescence polarization imaging include that it provides quantitative results in real time, it's cost-effective, and can achieve diagnosis in single cells without the need for extensive tissue processing. The fluorescence polarization method could be used to reduce incidence of indeterminate cytology, reduced costly molecular analysis, yield a definitive diagnosis at the initial fine needle aspiration cytology appointment, and to minimize unnecessary diagnostic surgeries. Here are recommendations for future directions of the fluorescence polarization technology. 1. Explore in vivo applications of methylene blue fluorescence polarization for cancer detection. As previously mentioned, one of the advantages of using methylene blue is that it is already approved by the United States FDA for in-human use. In addition, results of our cell viability experiments demonstrated that this method did not adversely impact the cells during or after imaging. In vivo applications could include using this approach to guide FNA specimen collection or to deliver the diagnosis in situ. 2. Examine methylene blue fluorescence polarization correlation with the tumor grade and molecular subtype, which may provide clinically relevant data regarding the disease prognosis and therapeutic response rates. 3. Investigate applications that obtain fluorescence polarization values through spectroscopic measurements, which may be a more feasible approach in low resource settings where spatially resolved measurements obtained through imaging could be prohibitively expensive. And 4. Develop rapid sample preparation protocols to facilitate widespread clinical adoption of fluorescence polarization technology for cytopathology. I would like to acknowledge Professor Anna Yaroslavsky, the director of the Advanced Biophotonics Laboratory at UMass Lowell, and Tyler Iorizzo. I would also like to acknowledge our clinical collaborators, including Dr. Ashraf Khan of UMass Chan Medical School Baystate, Dr. Dina Kandil, Dr. Andrew Fisher and Sarah Pantanod of UMass Chan Medical School, Dr. Leja Joseph and Thomas Ackerley of Lowell General Hospital, and our study statistician Alana Musikansky of the Massachusetts General Hospital Biostatistics Center. Funding from the sources listed on this slide is gratefully acknowledged. For additional information regarding these projects and many others, please visit the Advanced Biophotonics Lab website. Finally, I would like to thank Misha the cat, who has provided much support for these projects and many others.